and both my lab and I think this department as a whole are much the better for it. So I honestly think I could talk about Matthew for the full hour, but I don't want to cut into his time too much, so I'm just going to emphasize three things that I've really come to love about Matthew. The first is the breadth of his intellectual curiosity, both within science as well as extending to philosophy and the humanities and really all other aspects of life, and his willingness to engage in really fun discussions about pretty much anything. And I think this is something that is increasingly rare and just has been great to have as a part of the lab. The second is scientific, his creativity and rigor when it comes to the work he has done, and I'm sure the work he will do. I'll let him talk about the work himself, but you know, Matthew, it's easy to forget, came in here with virtually no wet lab experience, was extraordinarily interested in diagnostics, I think, following on his experiences in Africa, and developed an expertise in an area around cell-free DNA, and proceeded to both conceive and execute on I think, some really brilliant ideas in this space. You know, I was once told that when you finish your PhD, there should be one thing that you know more about than anyone in the world, and I think for Matthew, at this particular moment, that really is the case. Last and certainly not least, I'd like to acknowledge what an integral and positive member of our community Matthew has been, and I'm really talking about not just my lab here, but the department as a whole. There are, you know, I can point to some concrete things. For example, organizing courses like the philosophy of genomics that Maynard and I think Bob participated in teaching. The Dick Lewontin Journal Club, a very specialized journal club for Dick Lewontin aficionados. And stepping up effectively to serve as faculty at one point for teaching statistical genetics to his fellow graduate students. But I think as important as any of those more concrete things has been kind of the more under the radar, consistent and proactive role he's played in contributing to this and helping to maintain this remarkable community that is this department over these past years. So yeah, I really could go on for an hour. Matthew was really like I was trying to think of an analogy, and the only things I could come up with were an onion, many layers. Thomas Pynchon novel. But I'm going to shut up and let him talk. So just to explain the order of events, Matthew will have till the end of the hour to present the seminar, and then we'll take some questions from the audience, and then everyone is excused except for his thesis committee, which will stick around to ask him some hard questions. And there are food and drinks somewhere. Does anyone know where?
Third floor lounge.
Third floor lounge. And Matthew and the committee will join you up there as soon as we can. So, okay, Matthew. Switch over here.
Yeah.
Give me 1 second to recombobulate myself here. It'll take this time to let that moment sink in. Check. Is that volume okay for everyone? All right. One last thing. Great. Well, it's really great to see everyone here today. I'm very excited and a little nervous, honestly, to be up here sharing my experiences over the past 5 years, and hopefully I can convince you that some of the work I've done is meaningful and will have clinical relevance. And specifically, I want to talk to you about the work I've done looking at cell-free DNA, as Jay pointed out so kindly in this very lengthy and elegant introduction to me, where we've really tried to expand the accuracy, the resolution, and also the breadth of the things that we can look at with cell-free DNA. The search for biomarkers of disease is really as old as medicine itself. The art of uroscopy, or the examination of patient urine samples to learn about diseases that someone might have, dates back well over 1,000 years at this point. And here I'm showing a so-called urine wheel, where the colors of urine from a patient sample were matched to this wheel in order to diagnose a patient. Now, the urine wheel might seem a little bit quaint, and the diagnostic utility, it wasn't perfect, but nonetheless, it did actually have some success. For example, kidney disease was evidenced by a shift towards a dark brown color of the urine. And color wasn't really the only feature that proved useful. In fact, in the 1650s, Thomas Willis discovered that in individuals with diabetes mellitus, the urine had a pronounced a sweet taste owing to the glucose in the urine. Slightly later, Frederick Deckers discovered that in patients with proteinuria, the urine sample from that patient, when boiled with acetic acid, would yield a visible precipitate. So while medical technology has certainly improved in the 500 years since then, in some ways there remains this striking link to the past, as we can appreciate by looking at this colorimetric, this color change in a dipstick, which is still conventionally used to diagnose proteinuria. Biomarkers in blood have had a similarly long and checkered history, not always with the greatest success. Galen, who was a physician in ancient Rome, believed that diseases were caused by an imbalance in the four humors. By taking a sample of blood from a patient, he would wait for the blood to settle, and then essentially look at the different proportions of the four humors, what we would now call plasma, white blood cells, and different types of red blood cells. For example, jaundice was marked by an increase in the proportion of yellow bile. So while a large number of new and powerful biomarkers have been discovered in the blood since this time, nonetheless there remains a striking link to this past. The erythrocyte sedimentation rate, or ESR, shown here at the top, is a relatively common test for inflammation, performed by measuring the rate at which red blood cells settle during the course of an hour. Hematocrit, or the percentage of red blood cells in the blood, is an important diagnostic tool for anemia. So while diagnostic accuracy has certainly improved since these ancient times, both of these tests actually bear a remarkable similarity to early tests devised before the Cartesian revolution in biology sort of revolutionized the search for new biomarker discovery. And what a revolution this has really been. Today, an annual checkup at the doctor would likely result in what have been standard, would likely result in what are now sort of standard panels of blood tests, CBCs, a metabolic panel, a various enzymatic tests, blood chemistry. Now, these tests provide a lot more information than we can get just by looking at a tube of blood, and collectively they've had an enormous impact on health, both at the individual level, for example, in the context of detecting infections or heart attacks, but also at the social level, helping to guide, for example, federal dietary recommendations. Nevertheless, these biomarkers are still somewhat crude. The genomic era of the past 20 or so years has ushered in a push for biomarkers of precision medicine using these high-resolution biomarkers. So what goals might these biomarkers help us achieve? One of the key aims of precision medicine is understanding not just what broad category of disease an individual might have, but what specific features of that disease might be useful to design a customized treatment plan. Biomarkers are also useful after a diagnosis has been made, both for monitoring the burden of a disease and for detecting potential recurrence of that disease down the road after treatment. Let's look at two examples. The Philadelphia chromosome is a feature of many chronic myeloid leukemia patients in which sections of chromosome 9 and 22 are essentially swapped, creating a fusion gene called BCR-ABL, which is a hyperactive tyrosine kinase. The presence of this fusion is not only a sensitive test for CML, but also predicts the response to Gleevec. Similarly, about 20 to 30% of breast cancers are what's called HER2 positive, marked by overexpression of HER2, a cell growth factor receptor. Individuals with HER2 positive breast cancer may benefit from treatment with Herceptin. So these are undoubtedly useful tools for many patients, and are certainly compatible with this modern hypothesis that our genetic makeup explains at least a substantial portion of our disease risk. These are both for serious congenital disorders, as well as for complex traits, including lipid levels and macular degeneration. However, many of these emerging biomarkers require the use of invasive and uncomfortable procedures, like tissue biopsies, for example, and inpatient procedures in general. Ideally, what we'd like to find is a biomarker that's powerful, informative, non-invasive, and compatible with today's standard clinical workflows. Today, I'm going to talk to you about one such biomarker, cell-free DNA, which I've spent the bulk of my PhD working on. I'm going to try to convince you of the enormous potential of cell-free DNA in various different contexts. I just want to emphasize the word potential here, because while the stories I'm going to tell you today, I think, are promising, there's still much work to be done. Let me just briefly introduce the history of cell-free DNA. Almost 70 years ago, Paul Mandel and Pierre Mattei unexpectedly found nucleic acids, in this case both DNA and RNA, in the plasma of various individuals, both healthy individuals as well as a seemingly randomly assembled cohort of individuals with diseases ranging from tuberculosis to gout to obesity. What they were observing was a large number of molecules of DNA outside of cells, or cell-free DNA, which they could purify from the plasma of these individuals. These molecules represent the detritus, or the debris of cell death, the bits of the chromosomes that are produced when cells die and undergo apoptosis, and their components are broken down for recycling or excretion. This observation went, roughly speaking, nowhere. It was essentially an historical curiosity until the mid-80s, at which point there was a surprising discovery that in some individuals with cancer, a portion of that cell-free DNA appeared to derive from the tumor itself. The next decade, Dennis Lowe found evidence of fetal DNA, or actually what we now know more precisely to be placental DNA, in the plasma of pregnant women. These observations showed that different tissues, different organs were able to contribute to this pool of cell-free DNA within a single individual, suggesting that perhaps it could be useful for diagnostic purposes. So by this time it was clear that the cell-free DNA had diagnostic promise, although I'm not sure that even at that time the researchers quite appreciated the degree to which cell-free DNA would take off. Over the next decade, three key applications of cell-free DNA to clinical questions emerged. In particular, we can now use cell-free DNA to evaluate the health of a fetus during pregnancy, to detect or to guide the treatment of an increasing number of cancers, and to monitor the health of transplanted organs. Improvements to genomic technologies that are needed to interrogate cell-free DNA argue that I think additional clinical applications are on the horizon, although we're not quite there yet. Now, the genomics field has taken some admittedly deserved criticism in the past for over-promising. And we're clearly not immune to this even today. Recently, two prominent geneticists published an article in which they argued that cell-free DNA might actually be a stethoscope for the next 200 years. Well, I would suggest that the stethoscope is likely to be the stethoscope for the next 200 years. Nevertheless, I think it's clear that we've only scratched the surface with what we can do with cell-free DNA. And today I'll tell you three stories about how we might be able to improve the degree to which we can investigate various conditions with cell-free DNA. In particular, I'm going to tell you about approaches I've developed to improve the accuracy, the resolution, and the breadth of cell-free DNA investigation. And I'll start by telling you about how we can improve the accuracy in the context of pregnancy. Many women find themselves in a prenatal care setting, at least once during their lives. For some women, due to advanced maternal age or previous pregnancy outcomes, this can be a stressful experience, as they may be recommended for prenatal testing for serious fetal conditions, such as trisomy. Trisomy is a condition in which an individual has an extra copy of a chromosome, 3 copies instead of the typical 2. The 3 most common trisomies are known as Down, Edwards, and Patau syndromes, and collectively affect approximately 1 in 700 live births in the US. A series of increasingly refined and accurate screens have been developed over the years to identify affected pregnancies, including sonographic and protein-based screens. However, these screens still fall short of a diagnostic threshold, and so confirmatory procedures are needed in the event of a positive screen. While these diagnostic tests represent true gold standards, they nonetheless carry a small risk to both the fetus and potentially to the mother, and are certainly, even if not risky, quite uncomfortable procedures. And these include amniocentesis, chorionic villus sampling. So a non-invasive screening method that could achieve very high accuracy could hopefully reduce the burden of invasive follow-up procedures. Enter cell-free DNA. In 2008, two studies nearly simultaneously showed the possibility of detecting trisomy by sequencing the maternal cell-free DNA, representing a non-invasive screen whose performance characteristics were really on par or better than standard screening methodologies. How does this work? Well, during pregnancy, approximately 10% or so of cell-free DNA in the maternal plasma is actually derived from the placenta and represents the fetal genome. So by drawing blood from pregnant mom, we essentially have a window into the health of the fetus. But we have a substantial background of her own genome, typically carrying 2 copies of each chromosome. So we're trying to discriminate between 2 or 3 copies of a given chromosome in the fetus by sequencing the maternal cell-free DNA, and I'll try to describe how this test actually works. Imagine the cell-free DNA in mom's plasma as an enormous ball pit filled with different colored balls, where each ball is a molecule of cell-free DNA. In this example, each color represents a specific chromosome, and the number of balls from each color depends on how long that chromosome is. Longer chromosomes have more balls, shorter chromosomes, fewer balls. In the most popular testing methodology, the way the test works is essentially to reach in and scoop out 25 million balls, 25 million molecules of cell-free DNA, and looks at the colors by performing DNA sequencing. The goal is to identify whether there's an excess of a specific color, too many molecules deriving from a specific chromosome, relative to typical values in unaffected pregnancies. Conceptually simple, but technologically rather sophisticated. The growth of this class of testing has been remarkable. At least 4 testing providers now offer versions of this test in the US, with millions of such tests performed both here and abroad, leading some analysts to suggest that this is actually the fastest growing molecular test in the history of medicine. So how does this test perform? Well, one key criterion is the positive predictive value. In other words, the probability that in the event of a true test, of a positive test, that the fetus is actually affected with trisomy. We want this number to be as high as possible to minimize unnecessary follow-up invasive procedures. The challenge, though, is that the number of trisomic pregnancies is still rather modest, and this is especially a problem as the screening methodologies are increasingly applied outside of high-risk pregnancies to average-risk pregnancies. Because the true rate of trisomy is small, even a very small number of false positives can impact the positive predictive value in an outsized way. For example, to be more quantitative about this, one recent and high-profile study showed that even with false positive rates as low as 0.2%, which sounds like pretty good test performance, the positive predictive value of these screens nevertheless remained under 50% for all 3 relevant chromosomes, meaning that even in the best case, a positive test is still more likely than not to mean that the fetus is actually not trisomic. So ideally, we could reduce the number of false positive tests to increase the positive predictive value and reduce patient anxiety. The problem is that we still have an imperfect understanding of what causes false positive tests in the first place. Most examples of false positives are case reports and anecdotal descriptions of individual pregnancies, which offer little guidance to improve test performance and genetic counseling. With the help of a former colleague, Dr. Ronnie Simmons, I sought to fill in this picture. I, in particular, identified that maternal copy number variants play a significant role in false positive test results. I'll briefly explain what we found. A copy number variant is a region of DNA that a variable size that is present in a different number of copies of different individuals in the population. Essentially, a piece of the chromosome that's either duplicated or deleted. Some CNVs are medically relevant, implicated in a number of different disorders, but the vast majority are medically neutral, more or less benign. But a feature that all CNVs share, importantly, is that they change the amount of genetic material that is derived from a given chromosome. Remember, only part of the cell-free DNA is fetal, shown here in blue, and the vast majority in most pregnancies is maternal, shown in green. We're trying to distinguish whether the fetus has 3 copies relative to the mother's 2. If mom has a copy number variant that is a duplication, essentially what we're doing is stacking the ball pit with more balls of a certain color. And the degree to which that ball pit is stacked depends on the size of the CNV. If we have a classification threshold, shown here in red, the degree to which the ball pit is stacked may cause a false positive test. Here we see a surplus of maternally provided molecules, while the number of fetally provided molecules still represents fetal euploidy, as in this true negative case. To be a bit more rigorous about this, typically these tests work on the basis of z-scores, with a z-score of 4 or more resulting in a positive test. For a woman with no copy number variant, this probability is rather small, about 3 in 100,000. In the event of a copy number variant, though, the probability of a false positive can grow, mostly as a function of the size of the CNV. Larger CNVs, bigger impact. While I don't have time to go into all of the details, the key point that we can take home is that we can take published test metrics from these tests and use them to quantify, for a given copy number variant, what is the likely impact on false positive results. For example, a 500 kilobase copy number variant on chromosome 18 should lead to a roughly 700-fold increase in the likelihood of a false positive test for that chromosome, absent any other risk factors. Given this finding, we speculated that maternal CNVs may actually play an underappreciated role in this burden of false positive tests. To test this, we recruited four women locally, who had had false positive tests from a commercial testing provider. We obtained new blood samples from these women during the same pregnancy, although later in gestation, and processed each sample as though we were acting as the test provider. What we found was that two of the four women had large copy number variants, in both cases on chromosome 18. Case 1 duplicated over 1 megabase of sequence. This is a rare and quite large CNV. Case 3 is duplicated approximately 500 kilobases of sequence. While we're not able to say with absolute certainty that these CNVs cause the false positive results because we don't have access to the original testing data, our results do suggest that the increased likelihood of false positive tests caused by these CNVs makes this by far the most likely explanation. I next wanted to ask, to what extent would this finding possibly impact test results in the general population? To answer that question, in collaboration with Brad Coe from Evan Eichler's lab, I obtained a set of copy number calls, essentially copy numbers that exist in the population from approximately 20,000 individuals, and asked the question, how common are large CNVs? Essentially what we looked at was the interplay between the frequency of different sizes of CNVs in the population and the impact of test results, on test results that those CNVs would be likely to have. Given what I've told you already, this shouldn't be terribly surprising from a 10,000-foot view. Modestly sized CNVs, shown here as this blue line, are actually quite common in the population, affecting on the order of 1 to 50 individuals. However, they have a relatively small impact on the probability of false positive results, maybe a 10-fold impact or so, depending on when in gestational age the test is done. However, keep in mind that this test is now offered on the order of millions of women, suggesting that in aggregate these results may be substantial, while the risk for a single woman may not be significant. However, the more large CNVs, which are quite rare in the population, maybe less than 1 in 10,000 individuals, can have huge impacts on the probability of false positive tests. In particular, I found two key parameters that jointly determine the population level impact of test results. A technical parameter, the coefficient of variation of the test, which is sort of set by the testing providers themselves, and this CNV burden in the population. And the interplay between these two suggests that chromosome 18 is likely to be the most impacted of any of the 3 chromosomes, with lower impacts on chromosomes 13 and 21. Encouragingly, though, the findings that I presented here have already borne fruit. In our publication on this work, I proposed a rather straightforward analytical switch that testing providers could implement to essentially correct for this problem. In essence, hoping that simply by publishing the work, we would make our own work irrelevant moving forward. Indeed, Illumina, one of the top testing providers, recently reanalyzed some of their testing data in light of our publication and confirmed the role of maternal cell-free DNA in a subset of their false positive cases. They further described an update to their data analysis pipeline that should correct this problem moving forward. So the turnaround time from the publication of our study to its clinical impact was on the order of months. And importantly, I want to point out that the positive predictive values as a result have now been increased to over 50%. Still not perfect, but way better than under 50%, which is where we started. In this section, I tried to convince you that as non-invasive prenatal testing expands to lower risk cohorts, positive predictive values need to improve in order to keep the patient experience high. Maternal CNVs play a substantial role in impacting these positive predictive values, or at least they did until very recently. And by analytically accounting for their presence, we can boost test performance in a clinically relevant way. I'll now tell you a story about the ways to improve the resolution of cell-free DNA sequencing in prenatal contexts. Testing for trisomies, as I just described, involves a rather zoomed out view of genetic variation, looking for an extra copy of an entire chromosome. However, each chromosome actually consists of millions of base pairs of DNA, nearly any of which could vary across individuals. In some cases, in medically important ways. For example, we might zoom in a small region here on chromosome 12 to the gene PAH. Mutations in this gene, such as the specific splice site mutation I'm showing here, can cause fetal ketonuria, a metabolic recessive disease. It may be desirable, for example, on the basis of family history of disease, to design a test based on cell-free DNA for the presence of 0, 1, or 2 copies of this risk variant. We could apply the same strategy to the approximately 7,000 other medically relevant 7,000 other diseases with known molecular causes. The problem with this strategy, though, is that while many of these diseases are quite rare and perhaps don't warrant the investment of time for an individually validated test for each disease, collectively they impact up to 1% of live births. Designing and validating these individual tests simply is not practical. A single comprehensive test, on the other hand, that could simultaneously interrogate the risk for all such diseases may be preferable. What form would such a test take? Obtaining the whole genome sequence of a fetus, including both inherited and de novo genetic variation, represents one approach to this challenge. I worked with a former lab member, Jacob Kitzman, to develop a statistical framework for determining the fetal genome non-invasively, again by sequencing a maternal cell-free DNA. I'll briefly describe our strategy for doing this. I'll start by describing our approach to learning about the inherited variation, which is quite different from our approach to learning about the de novo variation. The first step in our approach was to determine the parental genotypes. In other words, what set of genetic variants is present in the parents and might conceivably be transmitted to the fetus. The reason for doing this is that it vastly simplifies the problem. Instead of having to make a prediction at every site in the genome, we reduce the problem space about 1,000-fold. We can ignore 99.9% of sites at the top where the fetal genotype is known a priori, and focus our attention only on the sites at the bottom where the fetal genotype is ambiguous. We do this by obtaining DNA samples from both parents and using standard methods for detecting genetic variation. The next step is to obtain a sample of mom's blood during pregnancy, in this example at 18 weeks gestation, and then to sequence the cell-free DNA from that plasma. The result of the sequencing is a set of what we call reads. These are short sequences of DNA that we can examine letter by letter, base by base. Remember, a portion of the maternal cell-free DNA is actually placental or sort of fetal in origin. In this case, in this study, approximately 13%, represented by the purple slice of this pie. The red background of this pie represents the maternal contribution to the cell-free DNA. So what should this purple slice look like? The composition will be determined by which of the 2 possible alleles the parent transmits to the child. Now, we don't have any way of separating the purple from the red molecularly, but nevertheless, we can still learn something about the fetal genome using statistical modeling. We can predict what the cell-free DNA should look like in either of these 2 events, where the mother transmits the A allele or the B allele, and similarly for the father. and then look at our real data to see which seems more likely given our expectations. So the third step then is to use that model to make our predictions. We know what the cell-free DNA should like in these two cases. Now what do we observe? We can march along the genome, making predictions at each site about the fetal inheritance, based on which of the two possibilities seems more likely. We do this first for the paternal genome, and at each site we see that dad is heterozygous, we make a guess. Here we're guessing that dad transmits B, here we're guessing dad transmits A, and so on and so forth for all sites like this in the genome. This actually works pretty well. It's a fairly straightforward strategy, but it yields almost 97% accuracy at these sites genome-wide. We then apply the same approach to the maternal genome. Essentially, which of the two alleles does mom transmit to the fetus? Again, we're guessing by comparing our expectations at each site to what we observe in the real data and choosing the more likely scenario. Well, that didn't work nearly as well. So what went wrong? The crux of the problem is really that there's too much sampling noise in the data at every position. If we could do, for example, 100 times more DNA sequencing than we did in this study, we might be able to use this kind of strategy to get the right answer most of the time, but that's simply not practical. So we need to find a better way. It turns out that the solution to this problem is haplotypes. Before we make our predictions, we need to add in a new step 3 to determine the parental haplotypes. What are haplotypes? So far, I've been talking about genetic variants as sites where there are two alleles, A and B, one on each chromosome. For example, this is one such site, there's another, and so on and so forth. There are many such sites on each chromosome. Haplotypes refer to the set of alleles that are jointly present on one copy of a chromosome. For example, the C at this first site at the top goes with the T at the second site and the A at the third site. Now, instead of talking about alleles A and B, what we can talk about are haplotypes A and B. The motivation for this is that each single base pair in the genome is not inherited individually from the mother, but rather chromosomes are inherited as large units with a small number of meiotic crossover events on each chromosome. Combining evidence across sites in the form of these haplotypes is likely to give us better accuracy. Ideally, what we would have would be a set of haplotypes that cover the entire length of the chromosome from end to end. But due to technical limitations that I don't have time to discuss today, we're not quite there yet. Instead, what we have are many haplotype blocks scattered along the chromosome, kind of illustrated here at the bottom. Less than optimal, but it's what we have to work with for now. So again, we're still making predictions based on the evidence that we see from our sequencing data, but instead we're combining evidence from multiple sites along a haplotype. Instead of looking at each site in isolation, we're now allowing for some noise in the data by saying, given all the different sites on this haplotype, which haplotype is more likely to be transmitted? How does this work? Well, when we apply this strategy, we're essentially modeling the fetal genome as a mosaic of the parental haplotypes. We get much higher accuracy, particularly for the maternal genome, where we started at on the order of 60-something%, and now we're up to over 99% accuracy. I'll now briefly touch on our approach for detecting de novo genetic variation, the 50 to 100 or so genetic variants that newly arise in each generation. So here, knowledge of the parental haplotypes doesn't help us because these variants are not present on the maternal haplotypes. They're present only in the parental germ lines. And I want to point out that while the number of these variants, the count of these variants is actually quite small relative to the number of inherited variants in the human genome, their medical relevance is likely to be outsized importance due to their outsized importance in dominant disorders and with severe phenotypes. So we have to get these right. That's important. So they're important, but they're really, really hard to accurately call. Why is that? Well, first of all, we have a much bigger search space, on the order of 3 billion base pairs. We're no longer able to restrict our view only to that 1/1000th of the genome that we used for the other predictions that we made. And second, they appear at very low frequency in the plasma. Here's an example of one such mutation where we see that the de novo allele, the C allele, appearing in 3 out of 93 cell-free DNA molecules that we examined at that position. Now, this might not be a big problem. I mean, after all, we do see these mutations. But the problem is that sequencing technology remains imperfect. sometimes the sequencing data that we obtain does not accurately reflect the underlying truth of the molecule. Let's say a DNA sequencing instrument has an accuracy of 99.9%, roughly in the range of today's technology. When we're reading millions of fragments in an experiment such as this one, that's still a huge number of mistakes that that the machine is making. And each one of them looks like a de novo mutation using this kind of strategy. In this study, we ended up with over 20 million possible de novo mutations using the strategy. And again, we expect 50 to 100 of these to be real. What do we do about this? Well, honestly, I have to be straightforward with you. The options aren't great. What we did in this study was to apply a series of filters to the data, attempting to remove the errors and leave behind only the true variants. Now, some of these filters are principled, based on known error modalities of the sequencing instruments, and some are rather ad hoc and kind of fit directly to the data from this experiment. The goal here is to trade a small reduction in the sensitivity in the number of true variants that we observe for a suitably large improvement in specificity. Nonetheless, this works to some extent, but we're left at the end with a sensitivity of 50%. We've lost half of the true variants this way, and we still have 4,000 false positives. Still not great. And we've tried machine learning approaches, which work roughly comparably, maybe a little better in some scenarios, but I would definitely be lying to you if I told you that I thought machine learning was the answer to this problem. Moving forward, robust and clinically useful detection of de novo variants is one of the major outstanding challenges in prenatal testing with cell-free DNA. And I think certainly one area where new ideas could have a very large impact. So despite this outstanding challenge, non-invasive fetal genome sequencing technology is now achievable. It has been successfully performed not just by our group, but by at least 2 other groups around the world, with high accuracy for predicting inherited variation, and as I mentioned, kind of crappy accuracy for predicting de novo variation. I do want to stress, though, that owing to the high resolution of the type of predictions that we're able to make using this technique, there are important ethical issues raised here, and, as well as implications for genetic counseling, for clinicians, for prospective parents. And unfortunately, these are outside the scope of this talk, but I'd love to engage any of you in discussion about these topics later on. Now I'll move on to the final story I'd like to share with you today, specifically about a new method that we have for improving the breadth of what we can look at with cell-free DNA. As I said at the outset, there are really 3 key areas where cell-free DNA has made clinical inroads. Most strongly in maternal fetal medicine, but also to a lesser extent in cancers and an increasing number of cancers, and finally in organ transplantation. In principle, we'd like to be able to use cell-free DNA to investigate a much wider variety of clinical applications. What limits the breadth of cell-free DNA investigation? Well, one key limitation is that these applications typically rely on sequence mismatches in the DNA between 2 or more contributing tissues, like in the example with PKU, phenylketonuria, that I described earlier. We look at a pool of cell-free DNA for molecules that come from a tissue of interest, a tumor, a placenta, a donated organ, and we identify those molecules on the basis of sequence mismatches, sequences that are unique to one of the other tissues. I'll give you an example of a clinical test that's actually performed today, albeit in a very limited way. In melanoma, we're interested in identifying mutations in the gene BRAF, which we could profile by reading the cell-free DNA sequence from cancer patients. One of the most common mutations is known as V600E. Individuals with that specific mutation may benefit from a drug called vemurafenib, designed to inhibit this mutant protein. At a given time point, what we can do is essentially to take a sample of cell-free DNA from someone and quantify the number of times that we see this mutated BRAF. In other words, how often do we see a red molecule in the plasma? But at the first time point, it looks like there's actually a pretty high burden of mutant BRAF, which maybe is not great for prognosis. Then at a later time point, after treatment perhaps, we take another sample and we see that the proportion of BRAF mutated alleles has gone down. So while in principle this approach seems pretty straightforward and seems like a good strategy, in practice it can actually limit the breadth of what we can investigate with cell-free DNA. Why is this? Well, first of all, not all tumors have the same set of stereotyped mutations. So, if we don't know what we're looking for, it's really hard to find it. It's kind of conceptually similar to the problem I described with de novo variation. Secondly, and I think more critically, sometimes the tissues that we're interested in don't actually differ at the DNA sequence level. The major source of cell-free DNA in nearly all healthy individuals is blood, myeloid and lymphoid cells, which turn over in great numbers every day, even in healthy individuals. And while these cells may differ in some cases genetically from cancer, they're likely to be genetically identical to smooth muscle cells from the heart, for example. If we wanted to use cell-free DNA to potentially identify smooth muscle damage in the heart, and thereby perhaps have an early detection of a potential heart attack, we need a way to figure out which of these cells, which cell populations are contributing to cell-free DNA. We can't use DNA sequence to do that. I'll explain the method that I developed in close collaboration with Martin Kircher to infer the different contributing cell types to cell-free DNA, and thereby potentially open the door to monitoring other diseases and conditions. To understand the method, we need a basic understanding of the way DNA is packaged inside the cell. When most of us think of DNA, we probably think of one of two things, either the sort of classic X shape that many of us think about chromosomes as, or maybe as a double helix, kind of at the fine scale. Today, though, we're going to talk about one of the intermediate scales, protein complexes called nucleosomes, often thought of as beads on a string. They help turn this long linear molecule of DNA into an extremely compact package that can fit inside the nucleus. And each cell has millions of these nucleosomes. So the reason that we care about this is that when cells die, their chromosomes actually get chopped up into small pieces by a variety of different enzymes so that the debris can be easily removed or recycled. The cutting doesn't happen randomly, though. The places where the chromosomes get cut are influenced by the locations of these millions of protein complexes that we call nucleosomes. In particular, the cuts typically happen adjacent to the nucleosomes in these naked DNA regions. The cell's machinery then eats away at the DNA, and here's where the nucleosomes play an interesting role. They have a protective effect. Essentially, they act as a type of shield for the DNA that's touching them. But each nucleosome can actually only protect a relatively small amount of DNA, on the order of, say, 150 base pairs or so. The DNA kind of hangs on to the nucleosomes, maybe sort of as a life preserver kind of thing, as the cell dies and the debris makes its way into the circulation. The reason that this is relevant is because we're pretty confident that this is the way that cell-free DNA is born. When we take a plasma sample and purify the cell-free DNA, we can pretty easily figure out how long the molecules are. That's a simple problem to solve. And in sample after sample, we see this consistent pattern, this consistent picture emerging. The fragments look like they were protected by this nucleosome shield. So we asked the question, could nucleosomes help us determine the tissues that cell-free DNA is coming from? To understand how this works, it's important to understand that nucleosome positions vary by cell type. Here I'm showing two example cell types, B cells and some type of kidney cell. And all I want you to take away from this cartoon image is that the nucleosomes in the B cells seem to have kind of a gap here, and there's this orange and this green protein that seem to occupy a position that would otherwise be occupied by a nucleosome in the kidney cells. Another way they can vary is in the spacing. So not only can the positions be different, but here we see in the B cells, there's a wider spacing between these nucleosomes, whereas in the kidney cells, the nucleosomes are spaced more tightly. The crux of our method is really involves solving these, or answering these three questions. Where are the nucleosomes in our blood sample, in our clinical sample from a patient? Where are the nucleosomes in the many tissues and cell types in our body? And which are the closest matches between our clinical sample and the various tissues and cell types in our body? Our model is that enzymatic cleavage, as I described, preferentially happens adjacent to protein-occupied DNA. And the cleavage events, which we can read out by sequencing the cell-free DNA, should be biased for the regions of naked DNA, this open DNA in between the nucleosomes. So we developed a rather simple counting metric that helps us to quantify the degree to which any given base pair in DNA is preferentially protected or preferentially cleaved. After paired-end sequencing of the DNA, we perform alignment for the reference genome, and we identify the endpoints of each fragment. One endpoint here, one endpoint here. We do that for all such fragments in the genome. We next march along the genome using a sliding window, depicted in light gray. We simply count the number of fragments, shown in purple, that span that window, in this case, 2. We subtract from that fragments that have one or more endpoints in that window, in this case, 6. So for this first window, we get a score of 2 minus 6, or minus 4. If a window is centered on a nucleosome, we expect a slightly different pattern to emerge. The nucleosome, again, is going to protect this region of DNA. So here we should see more fragments that span that window, in this case 6, and here we subtract 3. So the score for this window is higher than the score for this window, which is sort of offset from the nucleosome position. We apply this counting process genome-wide to generate a window protection score, or WPS, for each position in the genome. The high WPS values indicate preferential protection, as we can see here, where it's sort of centered on the nucleosome. And low WPS values here indicate preferential degradation, or extra cutting, essentially, that's happening in this naked DNA region. Finally, we apply a peak calling procedure to this WPS, essentially looking for high consecutive regions of WPS scores across some number of bases to generate a set of nucleosome positions. So our prediction is here, and it sort of matches up nicely with where the nucleosome was in the DNA. We do this genome-wide, importantly, to generate a very dense set of predictions. So I want to just take a brief second to show you what the raw data look like. I know not everyone will understand all the elements of this figure, but I think it's important to see the type of data that we can get out of this type of experiment. So what I'm showing you here is an example region on chromosome 9. And I'm showing a few key types of data that we can get from this experiment. Shown here in green is the coverage. This is basically a way of looking at how many molecules to be sequenced at each single position in the genome. So it looks like for the, some positions were up to about 100, for lower ones were down around 25. The fragment endpoints of those molecules are shown in orange underneath. And already just by looking at where the endpoints lie, you can start to see a kind of relatively regular pattern of biased cutting. Finally, in red, I'm showing the WPS. This is the score that we calculate as I described in the previous slide. And you can see already kind of a sinusoidal pattern emerging. Finally, we can see where our nucleosome calls are. Interestingly, this region is a DNase I hypersensitive site. I don't expect everyone to necessarily understand what that is, but the key takeaway is that it gives us an expectation that the nucleosomes at this particular site should have a particular kind of spacing. They should look a little bit different than at any other typical site in the genome. In particular, they should be spaced more widely at this annotated DHS than they would than they do at adjacent regions, for example, over here. And in fact, that's what we see. Not only do we see a bit of a disruption in the regular sinusoidal pattern of our WPS, but we also see a wider spacing of the nucleosomes around this hypersensitive site. Now let's zoom out and look at how we're able to do genome-wide. We called about 13 million nucleosome positions, which sounds like a large number, and in fact we believe is the densest nucleosome call set that's been produced to date. And importantly, it covers most of the genome. We think of this as kind of a snapshot of what healthy cell-free DNA looks like, a roadmap, if you will, of where the nucleosomes are in healthy cell-free DNA. So that kind of gets at the first question. Now we move on to the second question. Where are the nucleosomes in the many tissues and cell types in our body? Ideally, what we'd have is this nucleosome positioning map for every different cell type we're interested in learning about. We'd have one for all the different blood cell types, all the different organs that might potentially be damaged and feeding into this population of cell-free DNA. Unfortunately, we don't have that. Those experiments have simply yet have not been done. Maybe at some point this will happen, but we're not quite there yet. So we need to find a proxy, some other way of getting at this information about where the nucleosomes are. What kind of proxy do we use? Well, there are many ideas that one could come up with. The proxy that we ended up choosing was to use gene expression, the degree to which a given gene is expressed in a given tissue. Nucleosome spacing correlates with gene expression, albeit somewhat weakly. However, because we're doing this genome-wide over a large number of genes, even a weak correlation is enough to induce a signal. Secondly, gene expression profiles are cell type specific. So gene expression profiles in one cell are not identical to another cell. And yes, they vary a bit between individuals, but they vary probably more substantially in many cases across different tissues and different cell types. So we turn to a gene expression reference that I think is fairly rich. It's the Human Protein Atlas, and it contains gene expression measurements from 76 different tissues and cell lines. I mean, this is all kinds of stuff: lung cells, skin cells, brain cells, you name it. It's got it. It's not complete, but it's way better than starting with only a small number of cell types. So we don't have a direct measurement of where the nucleosomes are in the tissues and cell types in our body, but we can use gene expression to make predictions about where these nucleosomes might be. Now we have to answer this third question: some way of matching our clinical sample